planktonic state, meaning what?
It's where the microorganisms are free.
They're free swimming and looking for some place to hang out. And so they find a place to hang out. Usually this place to hang out is what? Some type of inanimate object, or, you know, they're in a water environment, right? And so they're looking for some place to attach to. Now here on us, we also have biofilms on our teeth and different other parts of our body. And that can occur on us as well. But if we're looking at it out in the wild, such as in a pond or a lake or something like that, or even in your water bowl for your dog. How many of you guys have dogs or cats?
Yeah.
Okay. How often do you change your water?
Twice a day. Every day.
Okay, she's a much better parent than you guys.
Well, my dog drives.
Huh?
My dog lives. My
Well,
cat.
yeah. I mean, my dogs normally drink up all the water before. Yes, because that biofilm will form on top of it. Yeah, you see, if you look at their water at the end of the day, you'll see a film on top. That's the biofilm. So these bacteria are basically looking for some place to hang on to. When you're looking at the biofilm that's in your dog's dish, usually they began sticking to the edge first, right? And then the biofilm grew out away from it. But they at least have something to attach to, okay? And so they're in a planktonic state first, right? You're free swimming, they find a place to land. What do they normally use to land and attach to it? Flagella? That's what they use to swim. Huh? Mattress? Only in Matrix.
Yeah.
They make that later on. So they may have outer coating on their cell. Remember we talked about the glycocalyces, slime layer. Slime layer that they have on them is conducive to making a biofilm because it makes them sticky so they can stick to a surface as well as stick together. Also, one thing they could be using to stick to the surface would be fimbriae. Fimbriae are also external features of cells that allow them to stick to a surface. And so once they found a surface to stick to and they adhere to that surface, then what do they do after that? The matrix is being formed. What's this matrix being made up of?
DNA proteins.
DNA proteins,
Fibers of polysaccharides.
polysaccharides. So basically things that are going to make them sticky,
Did you
cohesive
say what?
together.
DNA what?
Huh?
What did you say? Protein and what?
DNA proteins and proteins. Okay, so they're starting to produce this EPS, or this extracellular matrix that they have. And what does that allow them to do? Communicate how? Form sensing. And so this form sensing is sending out a chemical message to other bacteria, and you may get more than just one species joining this biofilm. So it's going to become more of a bacterial community. And so through form sensing, they're attracting other bacteria to come and form this biofilm. And once they start forming this biofilm, what else do they need to form? Channels. So they need an infrastructure within this biofilm in order to have free swimming bacteria swim through those channels, communication go through those channels with chemicals. Their waste may be funneled out of these channels. Nutrients may be funneled into these channels. So they're basically building a biofilm city, which happens to be the title of Cape Verizon biofilm formation. If you ever get a chance to read it, it's a page turner.
It's a page turner?
It's a page turner. It can do that too. If you're having trouble sleeping, then you can get a copy of that and read that for nighttime. which is asleep. So they form this infrastructure, they have channels, they become a biofilm city. What happens after this? Some of them will escape away from there to help form another biofilm somewhere else. It starts all over again. So as long as you have that information in your essay, you should be good.
So what is it that escapes? The biofilm?
Some bacteria from that biofilm will break off and escape and then form another biofilm some other place.
Okay.
Such as what happened in Haiti, right after they had their earthquake, and then they had the hurricane, and then they had relief workers come in from some country. I can't remember the name of the country. It's in Eastern Asia. Where's the Dalai Lama from? Tibet. Okay, so they were Tibetan rescue people. And the one thing you got to understand about vibrio cholera, because that was the outbreak that happened there, is each region has a different serovar of that bacteria. I mean, they have different species or subspecies of it. And genetically, we can find out where it came from. And so when they did the genetics of that strain of vibrio cholera, they found out that it was not indigenous, it actually was brought in from someplace. So they were dumping their waste into the water, and the vibrio cholera was forming biofilms, and as the water got disturbed, they broke up and they went downstream. As they went further downstream, they infected more people as it went downstream. All right, so that was a side note that you probably don't. Number 3, describe in detail the typical microbial growth curve and describe the events that occur in each one of them. Right? So, what does your typical growth curve look like? And what type of system are we talking about here? A closed system. So, that means there's no introduction of nutrients and there's no removal of waste. So, I put my bacteria into the plate, and they're at a state of what we call the lag phase. What's going on in the lag phase? They're adapting to their environment. Are they metabolically active? No. Are they actively dividing? No. So they are metabolically active. They may be growing a little bit in size to prepare themselves to divide, but they are not actively dividing.
That's called the closed system?
That's called the lag phase.
Oh.
The lag phase is when they are metabolically active, but they are not actively dividing, but they are maybe growing in size, preparing themselves to divide.
So they do have growth, or they have no growth?
They have no growth in population.
Okay.
Okay.
Like growing in size
So no growth and.
Yeah, because you know how some cells, when they divide into two, the cell has to grow in size a little bit to accommodate the invagination of the cell wall. If you were to take microbial physiology, you would spend a whole 3 or 4 weeks just talking about that.
So they're basically just growing in size to be able to divide?
To prepare themselves to divide. Yes. So, after this phase, they found their food, the carbohydrate source, and so they start going like that. That's an exponential growth, right? So this is what we call the log phase. If you know anything about math and you're doing any logarithms and those type of things. I hate them too. That's why I'm a biologist. Anyways, they're going through the log phase. What's occurring in the log phase?
Exponential growth.
Exponential growth. So at this point, there is an abundance of nutrients, and there is not that much waste, because when these things grow and divide and they use up that carbohydrate, they're going to be putting off metabolic waste. If you were to do a graph of metabolic waste production with this growth, it would look the same as that. And the thing is, you have 2 phases. Remember we talked about using up glucose and then maybe using lactose? Well, you have this other growth, usage or production of metabolic waste from glucose, it levels off. They find lactose, and they have even more metabolic waste from the lactose as it goes along. So this will be in your glucose, this will be lactose, okay? And at this point, they would stabilize, but then they would find another source of energy and keep growing. But in this case, we're just talking about your typical growth curve. So once they reach here, it levels off. Why does it level off? Because it's stationary. Your growth and death are equal. The amount of cell replication equals the amount of cell death. You're going to have, your nutrients are going to start to decrease, and your waste are going to start to increase. That metabolic waste can become toxic to bacteria and kill them. So, as you have those that are growing out and away from, you notice that when you put bacteria on a plate, their colonies grow outward, right? From that single point. Well, the ones that are in the middle are your older bacteria. And you'll notice that when they start dying off, they'll be dying off in the middle, but that outer portion will still be viable. Okay? So, you have equal amount of death as you do equal amount of growth. Your nutrients are starting to decrease, and your metabolic waste is starting to increase. Okay? So this will be your stationary things. Okay, so now you reach this point right here. What's going on there? This is your death phase. Okay, so you're going to have more death of cells than more growth of cells. And the reason why that is because the amount of waste has overtaken the amount of nutrients that's there. Okay, so
What did you say we have more what?
more waste. You have a higher accumulation of waste than you have remaining nutrition. And that nutrition is dwindling down very, very small. Okay? And so only the hardy ones are going to be hanging around. They may be the ones that change from using glucose to lactose and successfully, and they can continue on growing. But for the most part, there's more death than growth. The amount of nutrients is very, very low, and the amount of waste is very, very high.
Okay, wait, so what happened in the stationary phase?
In the stationary phase, we have equal amount of growth as we have the equal amount of death. The amount of nutrients is starting to decline, and the amount of waste is starting to increase. Question? Got that? Just pretend like you got it. Okay. All right. So number 4. Describe in detail the process of performing a serial dilution and a viable plate count for estimating microbial population size. What does that mean by that?
Is that like what we looked at in
You
lab?
need to dilute a liquid colony to get a better isolation.
Did we do that in the lab?
With the little squares that counted it? Is that what it is? Or
No, we didn't do it in lab. We just did it.
Okay. So, describe to me what a serial dilution is.
You need to dilute a liquid colony to get a better isolation.
Okay, so I have 10 mil of a bacterial culture here that I'm starting off with, right? Let's just-
Isolation.
And then I also have one that is 9 mil. Okay,